the various ways in which mutations can happen, we are now going to look at some of the features of mutations that make them interesting from an evolutionary biology perspective. So, just to clarify, here I am primarily going to talk about mutations happening at the ATGC sequence level. I am not talking about chromosomal mutations or whole genome duplications, polyploidy change, polyploidy and so on and so forth. So, the first feature of mutations is that many mutations are pleiotropic, which means that they end up affecting not one, but multiple traits. So, just to give you one example, most of you probably know this person. This is Peter Dinklage, a reasonably famous American actor who was in Tyrion Lannister in Game of Thrones. So, this person, as you know, he suffers from a certain physiological condition known as achondroplasia. So, in achondroplasia, what happens is that there is a gene called the FGFR3 gene. There is so people who have this condition, they have a mutation in that gene, and this mutation ends up interfering with the conversion of cartilages to bones. So, obviously, if anything affects your bone development, that is going to affect your stature, I mean, which is what we see in the case of Dinklage. But what it also does is it ends up affecting a huge number of other physiological functions. So, for example, people who have this condition, very often they have what is known as sleep apnea. So, some people, when they are sleeping, suddenly they feel short of breath and they wake up like this, that is sleep apnea. Many of these people also have recurrent ear infections. So, the reason I am highlighting this point is that very often when one talks about mutations, they talk about mutation as this mutation leads to that one effect. Okay, this mutation makes the plants taller, or they make the plant shorter, or they make the plant go from yellow to green. Now, that is because we typically end up focusing on one thing, but that does not necessarily mean that that is the only thing that the mutation is doing. Many a times, many mutations end up affecting multiple characters, and that is why the effect of a particular mutation, when you are thinking about its evolutionary effect as opposed to its simple physiological effect, you have to think in terms of all the traits. It is very evolutionarily it is not useful to think about it as doing just one thing, because most mutations actually do not do just one thing, they do many things. The second interesting thing about mutation is that many a times the effects of a mutation, there is a trade off. In other words, if it ends up benefiting you in one way, there are many cases where the mutations end up having harmful effects in other ways. So, just to give you one example. So, this is a particular frog which ends up eating poisonous toads, right. So, this is the genus called Leptodactylus, and because these guys eat poisonous toads, so obviously they need some way by which they can resist that poison. So, turns out this is a very interesting study by Mohammadi et al. I have given you the entire reference. So, turns out that these frogs, they have developed not one but 12 different amino acid changes or mutations in a gene called the ATP1A1R. So, the ATP1A1 gene, this is a very important gene for these organisms, because they end up maintaining the ionic balance across the membranes. Now, what these people did is, Mohammadi et al. Did is, they figured out that there are 12 amino acid changes, and then they ended up engineering the proteins with those amino acid changes and tried to figure out what functions those proteins are playing, and therefore, what is the role of each one of those amino acid changes. So, what they figured out was awesome. So, they saw that initially the let us say in terms of resistance to toxin, the organism was here and then when the first mutation happened, that increased the resistance to toxin quite a bit, but it also ended up reducing the enzyme activity. Okay, remember I told you this is a very important enzyme for the frog. So, that enzyme activity was substantially reduced. Then there was a second change which further increased the resistance to toxin, however, that again reduced the enzyme activity quite a bit. So, 2 amino acid changes which are reducing the enzyme activity, and then there were subsequent 10 other changes because of which the resistance level slightly increased, but what happens is that the enzyme level activity level increased quite a bit. So, this is how this organism was able to become more or less similar, the enzyme activity level remained similar, although the resistant level went up quite a bit. So, this is another example to show you that if you are thinking about what a mutation is doing to an organism, you can't just focus on one thing, say increase in the resistance to toxin or increase in the resistance to enzyme activity, increase in the enzyme activity and so on. You always need to integrate over all the effects that a mutation is having. So, third one, this is actually almost personal. So, in evolutionary biology, most of the time we say that mutations are deleterious. Now, here at IISER Pune, I have many colleagues, and one of them, Professor M.S. Madhusudan, he is a physicist and also a bioinformatics person. So, I was having a discussion with Madhu, and he said, yeah, most mutations are neutral, they do not affect the organism in any way. So, this happened a few times, and we realized that something is going wrong, because I was referring most mutations as bad and he was referring most mutations as having no effect. So, then we puzzled how can both be true, and we chatted a lot, and then it actually took us some amount of discussion to figure out that we are actually defining mutations in a very different way. So, for a structural biologist like Madhu, mutation is essentially a change in the ATGC sequence. Now, if there is a change in the ATGC sequence, then actually most mutations do not really have much of an effect on the organism. Why is that so? That is because, as I told you, many mutations they do not even change the amino acid. Many mutations they might change the amino acid, but they might change, let us say, a basic amino acid to a basic amino acid. Okay. So, it is like the same kind of amino acid that is being formed. Again, many of those changes actually have little effect. And thirdly, for many proteins, particularly the enzymes, you know that there is something called an active site, and then around that active site there is a lot of the protein backbone. So, any change that is happening in the active site obviously might end up having a change in terms of what it is doing to the organism. But if you are thinking about changes in the protein backbone, even if the amino acids that are being formed are of a different type, even if they end up changing the conformation of the protein slightly, that will probably not have much of an effect in terms of how the protein acts. That is why a large fraction of the mutations that happen in terms of changes in the ATGC sequence, in terms of what they are doing to the function of the protein, they do not end up doing much, which is why for structural and molecular biologists they define most mutations as neutral. However, evolutionary biologists who are trained like me, most of us think in terms of mutations, in terms of what effect they are doing to the organism. Okay. So, if there is no effect at the organismal level, we do not even call it a mutation, we do not even think a mutation has happened. Now, why is it that evolutionary biologists think this way? Remember, evolutionary biologists, we are studying mutations much before molecular biology came about. So, for example, for people like Hugo de Vries, who were studying mutations even before they knew about DNA or DNA structure or anything, those people they were studying in terms of the actual effect that was happening on the organism, right? And therefore, their conceptualization of mutation itself is a change in terms of the organisms, whether it is getting benefited or harmed. And that is something that evolutionary biologists have still continued, and if you look at it from that lens, then most mutations which actually cause a change, most of those changes are harmful, right? In that context, we say that most mutations are deleterious. So, is there a way to take care of or somehow bring both these definitions into one umbrella? That can be done. So, the simple way is to think about mutations is that most mutations will not affect the organisms, but the ones that will affect the organisms, most of them are going to be deleterious, most of them are going to cause harm. Okay. So, let us give you an example of what I mean by this. So, this is a study which was done on yeast. So, what they did was they took a certain protein known as HSP90. Most of you might have heard about this protein. This is a heat shock protein, and this ends up playing a role in terms of allowing the organism to survive, as the name suggests, temperature changes, high heats. However, heat shock proteins also have many other functions, and therefore, if you end up somehow perturbing a heat shock protein, you end up seeing all kinds of effects on the organism. So, what these people did was they took 560 different mutations and asked what effect is this having on the organism. Okay. In other words, is it benefiting the organism or is it harming the organism? And so, this is a slightly busy graph, I need to walk you through this. So, they had 3 different kinds of mutations that are leading to stop codons, these ones, the black ones, mutations that are synonymous mutations, these white ones, and non-synonymous mutations that are actually leading to amino acid changes, which are these blue ones. So, what they did was they scaled it in such a way that the one which did not have any mutation, that one is called 0. Okay. And then, so all the ones which are having mutations which are leading to bad effects, the deleterious ones, they are on the left side, and all the ones which are leading to a positive effect in terms of survivorship, reproduction or whatever, all those are on the right side of this line. Okay. So, the first thing that you see is that an overwhelming majority are actually on this side, the left side. There are very few which are on the right side. Okay. And on this side, the further you go from this 0 line, the more harmful the mutation is. Similarly, the further you go on this side, on the right hand side, the more beneficial the mutation is. So, the first thing that you see straight away is that the number of beneficial mutations on this side are way less compared to the number of harmful mutations. This is the main thing that I wanted to show you. And this distribution of the deleterious mutations, it is not uniform. Most of the mutations, they are causing a little harm. Some of actually majority of the mutations are causing no harm. Okay, that is why they are all on the 0, the peak is at 0. So, the mutations that are actually causing some harm, most of them are causing a little harm, a few of them are causing a little more harm, okay, these guys, and then as you increase the harm, the fraction which are causing that harm is actually going down, right. So, in other words, you have a kind of a exponential distribution on this side. So, the harmfulness of the mutation is actually exponentially falling off, right. So, the main point that I want to make over here is that because beneficial mutations are rare, a population might have to wait a very long time in order to get a beneficial mutation. On the other hand, harmful mutations will keep on coming. So, this particular point, particularly about most mutations being harmful, will become important when we talk about a kind of selection known as stabilizing selection, whose justification will primarily come from this diagram. The other thing I want to tell you is that particular distribution of fitness effects. So, this diagram where you have the fitness or harmfulness, beneficialness on x axis and the relative frequencies on y axis, this is what is known as a distribution of fitness effects or a DFE. So, the DFE of many organisms are actually of this type. Okay, overwhelming majority where things have been studied, it looks like that the DFE is of this type. Now, the fourth point, this is a very important point, is about what are known as somatic mutations and what are known as germline mutations. So, you know that in many organisms we have a germ soma differentiation. What is that? See, all organisms are starting as a single cell, right, the zygote. However, during the process of development in many organisms, early on in the development, a group of cells, they become the reproductive cells. These are the ones that are known as the germline cells, and the rest of the cells, they form the entire body of the organism. So, for example, when it comes to say humans, okay, all our entire body is essentially the somatic cells, whereas there are few cells. In the case of males, it is those cells which are leading to the sperms. In the females, these are the cells which are leading to the ova. These cells, they actually differentiate very early, during our embryonic development. Now, think about what is going to be the fate of a mutation which is happening in a somatic cell. So, suppose there is a cell in my hand which has become mutated. What will happen? In terms of its effect on me, obviously it will have an effect, okay. But in terms of its effect on my kids, it will actually not have any effect, because only the mutations which are happening in my sperm cells or somebody else's ova cells, those are the ones which are going to be passed on to the next generation, which is why only germline mutations are evolutionarily relevant. All somatic mutations are evolutionarily non-relevant, with a caveat. Okay, please appreciate that. This we are talking in terms of what is being inherited. However, somatic mutations can, and of course they do, contribute to the survival of the person, right. So, I mean survival and both reproduction. So, the germline often contains things, contains mutations, contains genetic variations which can have an effect on the somatic mutation rate. So, just to give you an example, we know that cancer is a disease of the soma, right. So, cancer obviously is bad for us, cancer, or for any organism for that matter. So, cancer ends up affecting your probability of survivorship and probability of how many babies you are going to produce. And therefore, many organisms in their germline have variation which controls the mutation rate of the cancer. So, just to give you one major example, we know that elephants, very large organisms, lots of cells, more the number of cells, more the chances of getting mutations and accumulating mutations. So, the elephants, they actually have 20 copies of what is known as the tumor suppressor gene TP53 gene, and these copies, the 20 copies, actually reduce the chances of the elephant having a cancer to 50% of us humans who only have 2 copies of that gene, right. So, even though we are talking about a problem which will happen in the soma, the germline cells can and do affect the mutation rates in the soma. I mean that is important. Without that, organism will suffer what are known as fitness consequences. They won't be able to survive and reproduce properly. So, the basic implication over here is that not all mutations are evolutionarily relevant. So, with this we are done with 4 of the 6 features that we wanted to do. The remaining 2 we are going to do in the next discussion. See you.